Hello, everyone. Thank you for joining us this evening for First Fridays Connected, the online version of our First Fridays series. We hope you are all staying healthy and safe. We hope you can join us again on June 5th for more inspiring and thought-provoking discussions and lively music. We would like to give a big thanks to our presenting sponsor, XPRIZE, and our media and event sponsors, KCRW, Bank of America, and Spaceland Presents. Please don't forget to join other First Fridays Connected programming this evening. At 6 p.m., tune in to the discussion on the future of design and architecture, moderated by Pat Morrison. 7 p.m. is Dino 101 with Dustin Growick and Dr. Nathan Smith. At 7:30 p.m., La Marisol will perform live. Finally, KCRW DJ Novena Carmel will kick off your weekend in our DJ lounge from 8 p.m. to 9 p.m. If you've already registered, you should have received an email from the NHM with the link to these programs, or for more information, go to First Fridays Connect page at nhm.org. Now it is with great pleasure to introduce our hosts for Secrets from the Vaults and our resident museum expert that will guide you through the hidden secrets and untold stories of the Natural History Museum of Los Angeles County. First, our host, Allie Ward, science correspondent and host of the beloved podcast, Ologies. Joining Allie this evening is Regina Wetzer, Associate Curator and Director of the Marine Biodiversity Center at NHM. Please welcome Allie and Regina.
Hello.
Hello.
There you are.
Nice to be with you.
It's great to be with you too. Thank you so much in advance for letting me pepper you with questions.
Oh, I'm looking forward to it.
And so tell me a little bit about your history in science. Were you always someone that loved science? Did you love little things, big things, land things, ocean things?
I love being outdoors. I grew up on a chicken farm and had much opportunity to be outdoors. So camping trips to the mountains, outdoors.
Mm-hmm. And now, how long have you been working in the field that you've been doing now? How long have you been working with marine organisms?
A very long time.
Yes.
I've had 2 stints at the museum. So this last one has been 20 years. So this has been a good run. I was introduced to things marine once I got to college, pretty much. And I had a faculty member that had taken us to Baja and the Gulf of California. And I was introduced to invertebrates in the marine environment there and was hooked for life.
Were you much of a swimmer before that? Were you a water person?
Yes, I was a water person. I did swim team in high school.
So let's say we go for a dip in the ocean, right? Maybe we're looking out for dolphins. Maybe we're looking for a whale spout. But we are absolutely covered in marine invertebrates, right?
Absolutely. They're all around and they're small. Most of them are small. That's what I've learned over the years.
Mm-hmm. And I also want to remind anyone who's watching, you can leave questions in the Q&A and we'll get to those too if you have any questions for Regina. But I would love to know a little bit about the biodiversity in marine invertebrates. Like, how many types are we talking? I can't even imagine.
Many.
Yeah.
Many. So there's about 33 to 35 phyla of different kinds of major body plans of animals. Mm-hmm. And one group is the chordates, one phylum that birds, mammals, lizards, fishes, and all those are part of. And all of these other ones are mostly small, and they have a really deep history. Mm-hmm. If I may, maybe I can show you a little bit of the, I brought a phylogenetic tree.
Nice.
Something I like to do.
Let's
because
see it.
I like to know how big my world is or what my scope is like. And this is a way I do this.
Okay.
So let me make it.
I hope you bring this with you to dinner parties, just like a laminated phylogenetic tree.
Oh,
come on.
My lab has, so let's share this. Here we go.
Ooh.
Okay. It's big and cluttered, but it's all of life. So,
oh my gosh.
Origin of life a long time ago here. So here's my tree and it's branching out. All the little branches on terminal are the animals and plants and everybody that's living with us today. Mm-hmm. So about, more than 4 billion years ago, we have the origin of life. Wow. The little blue group in here are the metazoa, are the multicellular animals. We've got
Okay.
fungi, we've got all kinds of stromatolites. This light blue over here, if my colleague Brian Brown's listening, those are the insects. But I am going to dive in this little box right there, and we'll just look at the metazoans for the moment.
Okay. And what is a
and-
metazoa?
A metazoa is any organism, is any animal that is multicellular.
Okay.
So many cells.
Cool.
So here we have the origins of the metazoa. So it's just shy of 1 billion years. So again, all of this diversity has happened a very, very long time ago. The radiation of mammals, the radiation of all of the vertebrates. So here are the entire vertebrates here, including the fishes, which is the large group, birds, mammals, amphibians, dinosaurs, everything that has a backbone is in this group. We would be like one little here.
That's us.
So all these organisms on this side and everything I've cut off to the right is animals without backbones. They've been around for a really, really long time. So they have amazing morphologies, and most of them are very small.
Wow. Okay. So vertebrates, a pretty small slice of that pie. Now, what are some of these that you studied?
These is just, again, I want to emphasize that since the topic of tonight's First Friday is design and architecture, that most things are really old. Most animals are very old, and most things are very small, very, very small. I picked some larger ones as exemplars here. Everybody's familiar with jellyfish. Yeah. They have been swimming the seas for almost 1 billion years. To the right here
remarkable.
Is a glass sponge. It has been around for even longer than a jellyfish. And again, is very, very unusual. This is an animal that has no tissues. It has persisted since the longest of all of it's the oldest of the animal groups. Anybody that has gone through a general biology class and has cut apart a planarian and watched regenerative powers, this is a marine flatworm that's colored down here on the right. And again, this is an animal that doesn't even have a complete digestive system and is an amazing hunter, predator. It's a hermaphrodite. It has both sexes. It can do all kinds of amazing things.
Why?
Go ahead.
I'm curious. Why is that flatworm so brightly colored? Is that a warning color?
It can be warning colors. Some flatworms are brightly colored. Some of them are drab colored. Most flatworms do like to stay out of light and are usually hidden, unlike some of the sea slugs that are easily confused with them.
And now this little critter, we see this in our gardens, and this is not a bug, is it?
It's not a bug. It is a crustacean. It
Crazy.
Crazy. It is this is a terrestrial pill bug. And when I first joined the museum many, many years ago, on my first stint, I came to work for a curator whose profession was an isopodologist. I knew not the front end or the back end of these animals. I was in awe of the collections, and I learned pretty quickly and definitely fell in love with them. And some of you saw the picture of the large Bathynomus, which is usually my go-to because it's easy to describe because it is so large and recognizable. But every youngster can describe and take hold of pill bugs that are in your backyards and all over the place. And I primarily like to look at ones that are in the marine environment, and they're much smaller than those too.
How much of your work has to use a microscope, and what kind of microscopes are we talking?
That's a good question. We definitely use microscopes. We'll use a dissecting scope, so we'll magnify anything from 10 to 60 times. Some of the body parts, we would do dissections. We'll go under a compound scope, and so we'll do it hundreds of times of magnification. We also have geek glasses, and those help us in the field because seawater and microscopes don't mesh too well, and they let us give a first ID to many organisms. But a microscope is very key and definitely very useful.
We have an anonymous attendee asked a question: how do you feel about eating seafood? And I was wondering if that roly-poly is a crustacean. Could you theoretically eat them like shrimp? Or like a crab?
Yes,
Okay.
they would be edible.
All right.
Some roly-polies, I mean, I guess if I was really hungry, they would be edible.
Okay.
I don't, yeah, I eat shrimp, but it's not my favorite.
Okay.
I don't think of it as a very sustainable kinds of thing, as there's other seafood that I will eat.
So you don't, in terms of your work, that doesn't prevent you necessarily from thinking of some things as a food source? You'd have to eat a whole lot of dinoflagellates, right?
Yeah, exactly. or plankton that would be, towed up.
Now, I'm sure that you've seen in the news a lot, the bioluminescent tide. Can you explain at all what's going on for people who have been hearing about that?
Yeah, so those are microorganisms, and they actually just produce light. They become so numerous at particular times of the year and particular kinds of ocean settings, water temperature and salinity and currents. And they're absolutely wonderful light show. They're fabulous. There's been some beautiful photos, and I hope folks actually got to see them. It's wonderful to be able to dive in them. It's everything just glows.
Now, do you have to go out and do field work for your job? Are you analyzing things that are already in collection? What's a day in the life like for you, behind the scenes in the museum, in the vaults, in all of the collections?
Sure. It goes in fits and spurts.
Okay.
I love to get in the field. That's my absolute favorite. I don't get enough of it. I think most of my colleagues at the museum will probably say the same. I get to work in the lab. I don't get as much microscope time as I would like. My staff and my colleagues get a lot more than I do. A lot of time is spent in writing grant proposals, papers, administrative things, networking. As biology moves forward into combining all kinds of technologies, everything from field work to environmental DNA and the analysis of that, it's larger and larger teams with all different kinds of skill sets, and learning how to communicate with one another. Those are major challenges.
I'm sure. And what about the smallest thing in your collection, in the vaults? How do you even catalog something so teeny?
Oh, we have the very smallest things probably. And again, our collections probably stop at about a 0.5 millimeter in length, but you can't even pick those up with forceps. They're not called tweezers, they're called forceps.
Okay.
So you'd have a little loop, and you would basically scoop them out of the seawater or out of the alcohol, or pipette them up and move them into a glass shell vial. And Cotton Stopper, we're all about keeping all of our specimens very, very safe for indefinite, and specimens can be hundreds of years old, preserved properly. So
we have an anonymous attendee wants to know if you have a favorite sea critter.
Do I have a favorite sea critter? I'm very fond of octopus. I love seeing them. They are just amazing in what they can do and how they can change their bodies and their color and how they communicate. And they're approachable. They just, having 8 arms, what could I do with 8 arms? That would be.
I would get so much done.
I would probably better, yes.
And Brendan Layden wants to know if you think that it's possible to use computer simulations to project your tree of life chart into the far future. Do you think that we'll kind of try and figure out where that might go?
Yes, that's a wonderful question, is to look into the future.
Yeah.
Some things are diversifying at pretty rapid rates, faster than we had previously thought. Some things are faring not so well because of the things that we're doing to the planet, or the way we're moving organisms around, and some things get displaced. But yes, looking back in the tree of where things have gone extinct. Somebody might have noticed that trilobite clade that has gone extinct, a group that was, amazingly successful, left a beautiful fossil record, and just didn't make it at all. Other groups that have diminished, and then things that have radiated, like a lot of the mollusks, a lot of the crustaceans, and the insects, which are derived crustaceans.
And, we have a few questions about those bioluminescent organisms. Again, I think it's on a lot of people's minds because they've been seeing pictures, especially if, you know, we haven't been outside a lot. Probably people are wondering what's going on down at the beach. And so Evie Serrano wants to know, do those bioluminescent organisms lead to red tides? And an anonymous attendee wants to know, is all bioluminescence made through the same process, like fireflies versus, you know, marine invertebrates? Does that ever come up in your work at all?
It doesn't come up in my work specifically, but I work with colleagues that is their primary interest. The microorganisms that become so numerous and the ones that contribute to red tide, they can be a whole variety of, again, I showed the animals, the metazoans. There are groups, these are single-celled organisms that cause this bioluminescence. Some of them can be incredibly toxic. And they make fish sick and kill them, or they make sea lions and whales ill. So they come in all kinds of shapes and sizes, and the mechanisms for the light production, I don't believe is necessarily uniform either. Again, they have been around a very, very long time and are incredibly diverse in how they make a living and do their thing and reproduce and produce light.
Mm-hmm. It's I can only imagine if you didn't know about bioluminescence, or the first time you ever see it. Do you remember the first time you ever saw a tide like that?
Yes, probably Torrance Beach as a teenager.
Yeah.
And it was pretty spectacular because it was running through the surf and just splashing like crazy.
And have there been any field expeditions that have really stood out in your memory as favorites for you?
Oh, many. I've had the very good fortune to have had the opportunity to travel extensively and work in the tropics. Again, I study roly-polies. The particular group I work on are mostly tropical, so I've gotten to be in the Indian Ocean and the Pacific. But probably some of the most memorable ones are my very first ones, my introductions in the Sea of Cortez in the Gulf of California, where again, there was not only bioluminescence that was augmented with large trumpet fish, which I had also not seen before, and seeing slipper lobsters, again, under shallow waters. And it's also disorienting when you have all this light you're producing because you're flailing and it's completely dark. So those are some of the most impactful memories.
Oh, how magical. Nathan C. wants to know a little bit about building a collection of sea creatures and how to do it responsibly. Is there a way to collect shells or glass sponges? Are there any laws to be aware of or any ways to do it ethically and responsibly? What do you think?
Yes, that's a very good question, and that's one that's very, very important to address. When we do collect specimens or we receive specimens to the collections, we do it incredibly thoughtfully. There's a lot of permits. The marine environment has many, many restrictions, many jurisdictions that need to be abided by. We can't just go out and pick things up. And I definitely discourage anyone that really wants to pick up that dead shell that, you know, if they really like those little hermit crabs on the beach, please leave the shells. When the hermit crab grows and needs a bigger shell, if all the shells have gone home, it won't have anything to grow into, and then it's going to become seagull food or fish food, and it's the end of the hermit crab. Oh. So please do leave things on the shore. Most things that you take home from the beach will stink and be nasty, and mom won't like them. So again, these organisms do not preserve well. We have special techniques of how we preserve them, or how we photograph them and record their life history in the field before we bring them into the laboratory. And then when we do, everything is fully documented exactly where, when, how, what permits, what authorizations. We even have permits to even just collect seawater for eDNA and sediment samples for eDNA. So it is very heavily regulated. You can observe wildlife, and it's a little less destructive in marinas and harbors where you can, obviously if you have a boat, you're scraping off all those lovely encrusting invertebrates. And those are artificial habitats that are created by man. I'm a little more relaxed about that. But on the shore, please be very thoughtful. Turn rocks back exactly in the same position that you found them. Pick up the trash. It's always easy to scoop it up into a little cup, examine the organism, put it exactly back in the same intertidal spot that you found it, so that the next person can see it and the animal has a chance to survive. Taking it home is just a bad idea.
So maybe you're encouraging the hobby of photography instead of
Photography
shell
is absolutely. And I showed that pill bug, which is the terrestrial pill bug that lives in everybody's backyard. It is also the icon piece that we just put in for an iNaturalist. So for the City Nature Challenge, we just developed a terrestrial isopod hunt that folks can participate in. If you go to the museum's iNaturalist site, you will find the Southern California terrestrial isopod project. And these isopods also catch a virus, so that's one of the things that we're looking for. And then here, closer to the shore, we've been focusing on taking photos of the marine environment, and not only animals, but algae count as well. Algae, which are not quite plants, but they're seagrasses and algae, and all of those contribute. And those are wonderful records and much better memories than taking something home.
So yeah, so perhaps take up a photography habit and get the iNaturalist app, and then help scientists like Regina, I kind of a catalog and say, hey, I saw this over here. I saw this over here. Everybody wins, and nobody stinks.
Nobody stinks, and we haven't destroyed a little animal's life.
Right. Oh, such a good message. And what about you? You said that some of the things that are maybe frustrating is grant writing, things like that. But what is your favorite thing about your job or your work?
No 2 days are alike.
Yeah.
I would like to think I can set my own schedule, but it becomes a little bit more regimented. But it's so eclectic. I don't think when I first signed up to be a biologist, I actually had an understanding or appreciation of like one has to be a jack of so many trades. I didn't realize I needed to wear so many different kinds of hats. You would, again, the kind of science that much of what we do is primary field work, which really is quite simple, is a bucket and a net and, a pair of beach shoes and you're set to go. But the different kinds of, the documentation, the photography, the communications, the science communication portion of it, the grant writing, the collaborations, all of those are just so many multifacets. And again, folks in my lab and undergraduates that have been in our lab, some of the biggest contributions have been of those students that are an art major or are a communications major. I can teach them the biology, but I encourage anybody that is interested in biology in general or in things in museums, any kind of interactions or how people want to learn. So there's many opportunities to contribute and be part of a really fun, exciting, and very, very diverse environment.
And we had another attendee want to know, what kind of projects are you or your team working on? What is happening for you guys at the museum right now?
So we are all working from home, and my team is very active. 2 of our team members are at home with microscopes, and they are sorting and identifying a very large backlog of organisms. So that's really helpful. And then we have team members that are working on various grant writing aspects. We've been working on various new project ideas and focusing on the local marine environment. And it's been just, really diverse.
And another attendee real quick wanted to know, there's a lot of press about the vertebrates that are endangered or threatened because of human influences. Are there similar lists for inverts? I know we hear a lot about charismatic megafauna, what's going on with the whales, but are there little critters that are also...
That is true. There are endangered invertebrates. Unfortunately, things that are small are not nearly as well studied as things that are large or are, size to the size that we are, humans are. And so they are rarely make a list. There are some terrestrial insects that are on the list. There's one terrestrial or freshwater pill bug, but otherwise in the marine environment, we don't even know who's living out there. For example, there is an isopod that lives on the rocky shore, right here in the harbor, outside the harbor on the Palos Verdes Peninsula, and it's an undescribed species. We know it genetically, we have identified it, and it's still unnamed. We can go out and collect on any single day, just here on the backside of Palos Verdes. We don't have to go far at all. And there's just hundreds of new species all the time that we're discovering, not only morphologically, but now also with all these DNA techniques that we're using.
I mean, stupid question, but do you ever get to name them?
Oh yes, we get to name them. It is fun. And there's many of them on the shelf that are still waiting to be named. And that is one of the, the little bit frustrating things. Some of the habitats are disappearing more quickly than we can get them named. And so we're using modern DNA techniques to identify them. And because you saw on the tree how many there were that were not birds, mammals, and fishes, and it's unlikely they'll all get names, but hopefully they'll at least get recognized as genetic units, and they're all worth saving because we can all learn from the design and architecture directions, lifestyles that they can share with us.
Well, I hope one gets named after you, not only because Regina's a great name, but also Queen. Like, I feel like there's got to be a little isopod with your name out there. I'm just saying, but these are some of the secrets of the vaults, that there are unnamed new species out there under that roof, which is so exciting. So thank you so much for being here and letting me ask you so many stupid questions. But what
they're
you
not
do is
stupid at all, and I enjoyed it very much. Anytime.
One thing I love about roly-polies is, do you know the word conglobate?
Yes.
Yes. It's
to roll into a ball.
I think about terrestrial isopods whenever I'm having a bad day, and I just think I just want to conglobate on the couch and just roll up in a ball.
Absolutely.
See you in 8 hours. Yeah. So I'm terrestrial isopod. That should be a verb in and of itself. Is I just want to go terrestrial isopod. Well, thank you so much for everything you do. Thank you all the participants who were here and who are here, who sent in questions. Also, do not forget to join other First Friday Connected programming. It's so good. Starting at 6 p.m., there's a discussion on the future of design and architecture, moderated by Pat Morrison. And if you registered, you should have already received an email from NHM with that link. And then for more information, you can go to First Friday's Connected page at nhm.org. There's also Dinos 101. That's going to be moderated by Dustin Growick, who's a friend of mine who's amazing. So there's DJ sets, so stick around. There's more going on, and we will see y'all in June. Thank you so much, Regina, for being here.
My pleasure. Thank you, Ellie. Bye.
See you next time. Bye.